support staff in the CNS than you do in the PNS. And that should make sense. First of all, there aren't as many neuron cell bodies in the peripheral nervous system as there are in the central nervous system. So in the CNS, you've got 4 different kinds of supporting cells, okay? And they each have an independent function. Well, we didn't used to study glial cells very much because we thought that they were basically space fillers because we knew the neurons were the ones that did all the interesting stuff. And we were foolish because they actually do a hell of a lot more than fill spaces. So these are the 4 that are in the central nervous system, the 4 CNS glial cells. You can also see all of them on this figure right here because this is CNS. So let's talk about the 4 categories. One of the categories you've already met, let's get that one out of the way. The one that is responsible for myelination when it occurs in the central nervous system is a what cell? Oligodendrocyte cells. So that means that this white matter in the brain is white because of oligodendrocytes. Got that? So basically these are insulating the cabling to keep the signals going. Okay, so that's oligodendrocytes. Let's meet a couple of other ones. The next one is called an astrocyte. Here's an astrocyte. Any idea why it would be called that? Star-shaped. Yeah, star-shaped, exactly. So the astrocytes are the ones that we saw, historically speaking, we saw them like holding on to things in the brain, and they looked like scaffolding, like they're just like sort of places to put things. So they really thought that they were just structural stability in the brain until they figured out that in addition to providing structural stability in the brain, and you can see one right here as well, what they also do is to control the blood-brain barrier. You guys heard of the blood-brain barrier? What the blood-brain barrier is both a physical barrier and also a physiological barrier to keep funky stuff that's in your bloodstream from getting into your brain tissue. And so what these guys do, and we're going to talk about the blood-brain barrier in more detail later, what these guys do is, for instance, maybe you're thinking really hard and your brain needs more glucose. They'll increase glucose transport into the tissue. Maybe you're generating too many waste products. They'll actually move more waste products out of the tissue. Maybe you took in something funky and you don't want it to get into your brain tissue. They'll shut down transport, right? So you have both a physical barrier and then a physiological barrier. So that's one of the things that the astrocytes do. The next type of cell is called a microglial cell, and then I'll pick this one up. So here's a microglial cell. You see that little guy right there? The microglial cell is the smallest of the glial cells, not surprisingly. And it takes care of the fact that the blood-brain barrier has both good points and bad points. Good points keeps funky stuff from getting into your brain the vast majority of the time. Bad points is that also means that white blood cells can't get into your brain tissue. So you don't have a security force on the inside. But you do, because you got little ninja microglial cells. And what those do is if anything funky gets into your brain tissue, they will whoop up on it, right? So this is your security force on the inside of the brain tissue. And then the last type of cell we're going to run into several times today. Make sure that you can kind of picture what we're seeing here. So these cells right here, do you see them? What those are trying to show you guys is, remember I told you your brain was hollow. You've got these hollows in your brain called ventricles. These guys are in the wall of the ventricles, right? They're called ependymal cells. Do you see that they look very similar to epithelial cells? They're not epithelial because they're derived from brain tissue, but that's why somebody decided to start them with epi, right? So the ependymal cells, here's what these guys do, and they're going to be important. What they do is they suck a bunch of the best stuff from capillaries and dump it into the ventricles of the brain. They basically are making cerebrospinal fluid, and I know you've heard of cerebrospinal fluid before. What is cerebrospinal fluid? It's really, really rich fluid that bathes the inside and the outside of your brain, and it's made by these cells sucking stuff out of capillaries. Specific stuff, right? All the best stuff. So they take a disproportionate amount of glucose and amino acids and vitamins and ions that you need for transmission, all that stuff. So these are called ependymal cells. We will see those again later today. And then the 2 PNS glial cells, these two, right down here, you've already met one of them before, because how do you form myelin in the peripheral nervous system? What's this cell called? Schwann cell, or what's the other name for it? Neurolemocyte. Yes? Neurolumocyte. Okay, so the reason that my radial nerve is white is in part because of the neurolumocytes, right? Okay, and then these guys right here are, remember I told you that you can have neuron cell bodies outside of the central nervous system. There's just not very many of them. And when you do, they need a little mini support staff. Their mini support staff are these glial cells that wrap completely around them and control access and make sure that they have the right coffee and get rid of their waste products and protect them, all of that kind of stuff. These guys are called satellite cells. Any idea why they would be called that? Yeah, exactly. They're like completely like orbiting the cell right there, right? So those are satellite cells. Make sense? Okay. Do you remember what you call a chunk of neuron cell bodies outside of the central nervous system? It's in a ganglia. So you typically find satellite cells in ganglia, yeah? With the neurons in the ganglia. So this is a ganglion right here. Okay, so those are our glial cells. We're going to talk about a few of them again when we get into the brain. Last little tidbit about glial cells is why the hell should you care about glial cells? Well, we already talked a little bit about the fact that sometimes your neurons go bad not because something is wrong with the neuron, but because something is wrong with the support staff. Multiple sclerosis, again, is when you have your nervous system demyelinated because of a mistake with your immune system. It's an autoimmune disease that actually mistakenly attacks the myelin sheath, and then, of course, you start losing sensory and motor innervation to the periphery, right? So that's a mess, and that's a neuron problem that actually was a glial cell problem, okay? Parkinson's disease is another one. Parkinson's disease is when you start having these muscle tremors because of this. There's this really deep portion of your brain called the substantia nigra, and it's supposed to relay voluntary motor information out, like a relay station out, but the glial cells, for whatever reason, we don't know why yet, the glial cells in that region stop supporting the neurons, and then the neurons stop functioning, and you start not being able to control your motor activity. And then the other thing about glial cells is that unlike neurons, they are not amitotic. They go through mitosis throughout your life cycle, and that means something about the likelihood of cancer. So what do you guys think? If glial cells are mitotic and neurons are amitotic, are you more likely to have a glioma or a neuroma? A glioma. And there's a bunch of different kinds of gliomas. There's glioblastomas, in which you get something, the glioblasts make glial cells, right? Okay. You can have an ependoma, you can have an astrocytoma. You have far more of those than you have neuromas. Does that make sense? Okay. So, and then the very last thing that we're going to do on this set of notes is just to talk about what happens if you damage nervous tissue. If you damage nervous tissue, it depends on what part of the cell you damaged, whether you get any regeneration, and then also whether it's CNS damage or PNS damage. Okay? So, generally speaking, what happens if you damage a neuron cell body? Is the damage permanent? Yes. Generally speaking, if you damage a neuron cell body, the damage is permanent. Whether it's a CNS damage, which is, of course, where most of your neuron cell bodies are, or PNS damage, because the vast majority of your neurons are amitotic. So what I'm saying there is, okay, let's imagine that I had a blood clot and a stroke, right? Reduced blood flow to my occipital lobe, that those cells are gone after they're gone. Does that mean that I can never regain the functionality? No, luckily, your brain has a tremendous amount of plasticity, and while I never get those cells back, I could pass off the functionality to a different portion of the brain, right? Is it guaranteed? No, but it's not impossible that I could do that. And actually, women are slightly more likely to regain functionality after stroke than males are, for reasons I'll tell you guys later. Go ahead.
Is that kind of why, like, if you're a thief and, like, you cut, like, half of the brain out, you would do a half brain?
Uh-huh.
Yeah. So, like, that way, like, your other side of the brain takes over that part.
Yeah, it's far more likely to happen in a child. The regaining functionality is, you've got tremendous amounts of neuroplasticity as when you're really young that you sort of slowly lose as you age. So the younger you are, the more likely you are to recover from damage. Even if you lose a huge chunk of your brain, you can recover from damage. There are actually people with half of their brain that function normally, but only if they had half of their brain from the time they were teeny. Yeah? Okay. Now, so if you damage the neuron cell bodies, regardless of where, you have lost the cells, you may regain the function, the possibility of regaining the function. What if you damage not the neuron cell bodies, but the axons? Okay, so remind me, if I damage axons in the central nervous system, I am damaging not a nerve, but a what? Tract. Remember, it's called a tract, okay? But if I damage my radial nerve, yeah, I'm damaging a nerve. Okay, does that make sense? Okay, so let's see what would happen differently in those two situations. So if I damage my radial nerve, you guys won't be at all surprised, even if you haven't learned it yet, that the radial nerve, not surprisingly, will run down the lateral part of your arm and does innervation, both motor and sensory innervation, distal and lateral. So you might, for instance, control this activity. Does that make sense? So I want you to imagine that I damage my radial nerve now. Okay? So what is going to happen if I damage my radial nerve? But there are no guarantees, but let's look at what could happen. So in the peripheral nervous system, if you damage a nerve, you're damaging axons, correct? You're damaging axons. You're not damaging neuron cell bodies. They're all the way up here, right? So what's going to happen is if I damage a nerve, I would have damaged multiple axons, right? Because you've got multiple axons. So let's just look at one axon and see what could occur. So here's a single axon. This, if we were talking about the radial nerve, this neuron cell body would be up in the cervical region of my spinal cord, right? But I'm damaging it down here. You with me? So I'm damaging this axon. So what happens when you damage the axon is the region distal to the injury will just degenerate because it's cut off from everything, really. It's cut off from the neuron cell body. So you lose that. So I want you to imagine that this is a somatic motor neuron, so this is skeletal muscle, okay? Imagine that's skeletal muscle, okay? And I'm trying to control this activity. So the proximal portion just degenerates, but if you're in the peripheral nervous system, what will happen is those neurolemocytes will try to say, I got you, bro, and they'll try to help. So what they do is they start to go through cell division and they create a regeneration tube or a growth tube for the regenerating axon to grow into. Does that make sense? So you've got this little bud right there that's budding off of the axon, still connected to the cell body so it can still grow. And it's going to start budding and trying to make it into this tube. And we figured out just a few years ago, not me, but people figured out a few years ago that in addition to providing just a physical location to shoot for, it also, the regeneration tube does something called chemotaxis, in which it releases chemicals that are attractive to the growing, does that make sense? To the growing axon bud. So they make it more likely that you are going to hit the tube. Now, remind me, since this is skeletal muscle, right? What is true about this muscle unless and until you actually reinnervate? Can those muscle cells contract until you reinnervate? No. Remember, skeletal muscle, you have to tell every single cell what to do. So you've got paralysis of everything that was innervated by that particular axon unless you get regeneration. So, for instance, that could result in complete paralysis of this muscle, or it could result in weakness. If I only cut a few of the axons, you'll get weakness. If I cut all of them, I get paralysis. Does that make sense? Okay, so what will happen is as you grow, the remyelination occurs, and you can, although there is no guarantee, you can get complete reinnervation of the tissue. It is really slow, though. These axons grow about 1 to 5 millimeters per day, which sounds really fast, but that's going to have to grow all of that distance. And what if I damage my sciatic nerve and I'm trying to reinnervate my foot? It could be years before I know whether I get reinnervation. Does that make sense? So it could completely reinnervate, but sometimes they don't all hit the tunnels, right? Sometimes some of them hit the tunnels and you get weakness, but you still got activity. So it takes 1 to 5 millimeters per day. Now, has anybody in here ever had nerve damage? Yeah? So what does the doctor tell you after you have nerve damage about your likelihood of regaining functionality?
Either I would or I wouldn't.
Yeah.
Find out in a few months.
Find out in a few months. Sometimes you find out in a few years, depending on how long the axons are that you're going to regenerate. And really what you have to do is you just have to wait and hope that they hit the tube. So what does the surgeon do in this situation? So what if, you're in an accident and you've got like the two ends of the nerve that are actually widely separated from one another? What the surgeon will do is put them together, and the best that they can hope for is lining up the fascicles, right? Because everything else is microscopic, right? So they line up the nerve, they hope they line up the fascicles, and then they just say, now it's you. Do what you can, right? So it's not as if the surgery does nothing, but it's not going to do that microscopic reconnection. Okay, now what happens if you do the same thing in the CNS? In the CNS, if you do the same thing, you're kind of screwed if you damage white matter in the central nervous system. Because if you damage white matter in the central nervous system, these oligodendrocytes just pull up and they go, I'm out. And then what happens is the astrocytes will move in and they form the equivalent of scar tissue. Really, because if the astrocytes move in, even if I was trying to reinnervate, do you see that there's a physical barrier for me trying to reinnervate? So astrocytes move in to fill the space. They make a scaffold, but they have interrupted the likelihood that you will reinnervate. Does that make sense? So basically, CNS white matter damage is almost always permanent, just like CNS gray matter damage. Okay? So now, they have been working in the lab. It's still only in laboratory. I don't think it's even in animal models yet. They have been working at making regeneration tubes for the central nervous system that are coated with chemotaxic chemicals that will allow something to grow into them. Hasn't been used in humans at all yet. There is one treatment that has worked in humans, and that is embryonic stem cell treatments. So interestingly, embryonic stem cells, first of all, let me dispel some myths and talk about why a bunch of the research doesn't go on here. So when George Bush II was president, he got confused, as he often did. I'm from Texas. I know enough about him to be critical, okay? So what he did is he got confused and thought that embryonic stem cells came from aborted fetuses. And they don't, so I'll tell you where they come from. And he removed all federal funding for embryonic stem cell research, which meant what happened to our embryonic stem cell researcher scientists? They left the United States, the vast majority of them. So there's very little research going on in the United States, comparatively speaking. So here's what embryonic stem cells are and where they come from, and then here's how they've been used in this situation. So if I'm having fertility problems, one of the things that, if I'm relatively privileged, I can choose to do is to do in vitro fertilization, in which I take a drug like Clomid, and it makes me hyperovulate, and they harvest my eggs, right? And then they do fertilization in the laboratory from wherever I get the sperm, right? And then they let them grow up to about 32 or 64 cell stage. You're still microscopic. You are not a baby. And if this were in me, they would still be in my fallopian tubes. They wouldn't have been planted yet. Does that make sense? So this, if you get them too late, they actually won't implant if they shove them in there. So then they shove them in there and try to get them to stick. It's $10,000 a pop to do in vitro fertilization. It has about a 20% success rate. So because of that, you will hyperovulate and fertilize multiple eggs, not one, right? So maybe I had 25, right, from the process. Even in Utah, ain't nobody intended to have 25 children, right? So you shove them up there, either they stick or you give up, right, whatever happens. And then what do you do with the 15 that are left? Well, now you have these embryos that are frozen, and you can actually donate them for research. You could donate them to another couple. You could donate them for embryonic stem cell research. So now I have this embryo. It is not a baby. It is not to the stage that it would have been planted yet. These are what can be used. And they have to be really early because they have to be before differentiation, before they start deciding what kind of cell they do. So these can form any cell in the human body, and they've had success with spinal cord injuries. If you can get the embryonic stem cells in there before the astrocytes move in, then they actually figure out what needs to happen there. And they actually start the remyelination process and the repair process. And so people who have had embryonic stem cell treatment immediately after a spinal cord injury, I think you have like 36 hours, which doesn't happen in this country anymore because you can't fly somebody with a spinal cord injury to Israel. You just can't do it. So they've had success. Christopher Reeve had embryonic stem cell treatment, but it was years after the fact. But he actually still regained some sensation. with embryonic stem cell. In rats, when they started doing it, before they started using human models, they completely transected a rat's spinal cord, a group of rats, so that the rat was paraplegic, like lost complete sensory and motor innervation to the hind limbs, right? And then they shoved rat embryonic stem cells into the area, and they had about 60% regeneration of functionality within 4 weeks. So it's an interesting one. Of course, it's like gotten all politically whatever, but it's not a fetus. I always find this really interesting conversation because there are people who are really, really pro IVF, but really, really anti stem cell, and it just doesn't seem like a consistent position to me, right? It just doesn't seem to me, but you vote your conscience, not mine. Okay. This one, by the way, doesn't even really fall along political lines. Orrin Hatch was one of the biggest supporters of embryonic stem cell research in Utah, and that's probably the only thing I agree with him about. Okay, so let us now move on to brain. Note to self. This is not lectures that you want to miss. It has taken me 15 years to teach how to figure out how to teach the brain. You can try to learn it on your own. I don't think you'll be very successful. It's really hard. It's a hard story. It's got a bunch of unfamiliar terminology. So I would like for you to let me help you with it, help you organize it. Okay, so we are going to start on the brain and cranial nerves, and this is the last set of notes that will be on your next test. Okay, sometimes I don't test on the cranial nerves on the test. We'll decide on Thursday to see how far we get. So before we actually start getting into the structures of the brain, the first thing that we have to do is talk about how many different ways the brain and the spinal cord are protected. Because we just learned that if you damage them, you're kind of screwed, right? You are not getting the tissue back. You might regain some functionality if you're incredibly lucky. So let's talk about the brain and the spinal cord. So basically you have multiple layers of protection around the brain and the spinal cord. First thing you have is bone, right? You got your cranium. It is really freaking thick. Your cranium is really thick. So here's this a creepy, cool picture. Here's how thick the bone around your cranium is. Okay, and of course, around your spinal cord, you have bone, but not as much, right? Okay, and you've of course got spaces where it is not completely encased in bone because you need mobility in your spinal cord. Because of that, which one is actually more at risk, your brain or your spinal cord? Yeah, spinal cord is more at risk than your brain. So your brain is relatively protected. It's pretty hard to get through that bone, not impossible though. So your first line of protection is bone. And then the next line of protection are a bunch of envelopes of connective tissue that go completely around the brain and the spinal cord. Those are called the meninges. Singular is meninx. So singular is phalanx, phalanges. Yeah? Meninx, meninges. Okay, so you are going to have 3 different layers of these that go completely around your brain and your spinal cord. So I'm going to draw you a bad picture first, and then we'll start looking at the good pictures. So the bad picture that we are going to look at is, so if we do a lateral view of the brain connected to the spinal cord, I want to introduce you to the meninges as they go around. We have 3 of them. We have one that's snuggled up to the surface of the brain. We've got another one that's superficial to that, and we've got a really, really thick one that's superficial to that, okay? So the one that's snuggled up to the surface of the brain is actually stuck to the surface of the brain. Let me show you what it looks like. So it is the reason the brain looks shiny. So let me show you. This is a brain surgery picture right here. Do you see how the brain looks shiny? It almost looks like it has a serous membrane on the surface. It's not serous, but it's kind of the brain's equivalent to a serous membrane. So this shiny on the surface that has teeny tiny blood vessels underneath it, that is stuck on the surface of the brain. So imagine I'm drawing it stuck. I just can't draw it stuck, and actually, how do you see it? So this one, stuck to the surface of the brain and the spinal cord and continuous all the way down. This is called the pia mater, which means gentle mother. And you can't get the pia mater off the brain unless you know what the hell you're doing. It will actually pull the brain tissue off of it. So when you guys do the sheep brain dissection next week and you're looking at the sheep brain and it's shiny, you're looking at the pia mater. Okay? Just like if you were looking at the liver, you're actually seeing the visceral peritoneum when you look at the surface of the liver. If it's shiny. Does that make sense? Okay, so the next one out is kind of a weird one. It's hard to get a good description of that one, but I'll make it happen over the course of today. The next one you're kind of seeing right here, and it's spider webby. So this guy right here, the next one out, is really like a spider web. I'll draw it better in just a second. This is called the arachnoid, which implies what to you? Spider. And sometimes it's called the arachnoid mater, the spider mother, and sometimes just arachnoid. I'll draw the spider webby part in just a second. Okay, and then the most superficial one is completely opaque. If it's on the brain, you can't see the structures of the brain. So let me show you that one. Do you see this one? That one is called the dura mater, which literally means tough mother. And it is a tough mother. It's hard to get through it. So the dura mater is the most superficial one. And then you're going to have some spaces that will just define between them. Does that make sense? Okay, so if you are looking at the surface of the brain and you can see any of the hills and valleys, the dura mater's gone, right? If you are looking at a brain and it's like in an opaque sac, then you're still seeing the dura mater, okay? Some of you guys, when you do the sheep brain dissection next week, will get a sheep brain that has the dura mater on it, which is really cool because you can like pull it off and you can see all the stuff. Those are really expensive though, so we don't get tons and tons of them. Okay, so last thing is that there are actually spaces, and then we'll start going through the meninges specifically. There are spaces actually around these. Okay, so the dura mater around the cranium is actually stuck to the skull, so there's no space on the outside of this. So keep that in mind. But the dura mater around the spinal cord needs to wiggle. So what we need to have is a space on top of the spinal cord, which is a space that you have heard the name of before. See that space right there? It's usually filled with fat and loose connective tissue, and it's the space that is superficial to the dura mater. It is called the epidural space. Does that make sense? Okay. You don't have it around your brain. Your brain's not wiggly on a good day. Your spinal cord is wiggly, right? So it's basically, what's the fat doing? What do you think? Cushion, padding, protection, right? Because although your spinal cord isn't wiggling around a whole lot, your vertebral column certainly is. Yeah? Okay, so that's the epidural space. You do not have an epidural space up here. We'll talk about what it looks like up there. Okay, now we're going to start moving in. You ready? There's a space that is deep to the dura mater, but it's tiny. It's basically just a little teeny space with a little tiny bit of room underneath it for a little bit of fluid. There's a couple of little blood vessels in there. Have you ever heard of like a subdural hematoma? Yeah, or subdural bleed. It's because there's a few blood vessels in there, but there's not a whole hell of a lot of anything else. So this space right here is called a subdural space. Okay? So let me show you this figure. We're going to be looking at this figure multiple times today. Actually, I'll show it to you in just a second. I've got one more space to show you, and then I'll show that one to you. Okay, so now this next space is kind of weird. This next space is called the subarachnoid space, but I want to tell you that it's actually not sub, it's actually the intra-arachnoid space. So stick with me for just a second. You ready? Okay, so what the arachnoid is really a bunch of spider-webby collagen fibers that really attach the pia mater almost up to the dura mater. So instead of the arachnoid being one layer, it's actually kind of layered like this. Does that make sense? Okay, I'll show you a picture of it too. So the subarachnoid space is actually inside the arachnoid. So let me show you this. Ready? Okay. So, do you see this? So I want to find the layers real quick. You ready? Okay. Here's the cerebral cortex. That's the outermost portion of the brain. Then the white on the surface of that is the pia mater. Yeah? That's the shiny surface of the brain. Superficial to that is the arachnoid, but within the arachnoid is the subarachnoid space. So I would call it the intra-arachnoid space if they let me, but I didn't get to name it. So it really isn't sub. It's really within the arachnoid because you've got little spider webbies, right? So this whole layer is the arachnoid. Now, where is the subdural space? It's just that little teeny tiny space right there, just superficial to the arachnoid. Do you see that? Teeny tiny little space. There's sometimes just blood vessels running there. And then I run into the dura mater. Do you see? Okay. So the subarachnoid space is really within the arachnoid. That's where it is. And it's going to be really important because that's one of the locations that cerebrospinal fluid will circulate. Okay. All right. So now that we have all of our spaces and everything, let's start looking at them individually. So first off, let's talk about the epidural space. Now, I know you guys have heard of epidural injections before, right? So let's talk about what the hell you're doing when you have an epidural injection. When you have an epidural injection, you're trying to inject anesthetic, but you're not trying to go through the dura mater. Because you're actually not trying to anesthetize the CNS, you're actually trying to anesthetize the PNS nerves that come out through there. Does that make sense? Okay, I want to talk about that for just a second. Multiple reasons that an epidural could be done, but one that you're familiar with is, of course, childbirth, right? You're going to have an epidural injection, and you're going to learn these things in detail later, but I will tell you that the nerves that innervate your pelvis and your lower limbs come out at the lumbar region of your spinal cord, okay? So if I do an epidural injection, what I'm going to do is inject an anesthetic there, and it's going to diffuse out through those nerves that are coming out of the lumbar region and hopefully anesthetize, right, my abdomen and my pelvis. But is there any way that you can anesthetize the sensation of the abdomen and the pelvis without also anesthetizing the motor activity? So what happens during an epidural? Of course, you deaden the sensation, but what, can you move your legs? No, you usually can't move your legs, at least not the same way that you used to be able to. Because all of these nerves, remember guys, are mixed in function. Yes? Both sensory and motor, okay? And an epidural is not an exact science, just so you know. You're just going to squirt it into the tissue and let it diffuse out. And so if I lay on my right side, I'll get far more anesthetic on the right side, a little gravity, helping diffusion. If I put my butt up on a pillow, I can anesthetize myself all the way up to here. So that's what an epidural injection is. It's just trying to go into the epidural space. And of course, you would never do it up here anyway, right? There is no epidural space up there. All right, so that's an epidural injection. And then let's go into the dura mater. Okay, the dura mater is the tough mother. It's the most superficial. It's also the thickest of the meninges. It is like a super duper thick, dense, irregular connective tissue. And it is hard to puncture on accident. Okay, so so the dura mater, if you are around the spinal cord, the dura mater, you're actually seeing it right here. This dura mater is the white just deep to the fat. That's the dura mater. Around the brain, the dura mater is more complicated. So you got to try to listen to this. It's going to be a little hard to grasp until you see it 3D in lab, but we'll give it a shot. Okay. Around the brain, the dura mater actually has 2 layers. Okay, so around the brain, let me show you this figure real quick and then I'll show you another one. Okay, so around the brain, what you are going to have, let's look at this figure. Okay, so what we have is the subarachnoid space and the arachnoid are represented as gray. Do you see that? And then this white line right here would be one layer of the dura mater, and then the other white line would be another layer of the dura mater. The dura mater around the brain is double layered. The first layer, when I take a brain out of a cadaver, the outer layer of the dura mater is called the periosteal layer because it stays stuck to the periosteum. Okay? So when I take the cranium off, the periosteal layer would be on the inner surface and I would have to peel it out of the inner surface. So it stays with the cranium. Does that make sense? But when I look at the brain itself, this layer that you're seeing, that's called the meningeal layer of the dura mater, and that stays with the brain. Does that make sense? So you've actually got 2 layers. What is between them? Ready? So let's look up close. Ready? We're going to blow this up. Same picture that we looked at before. Okay, here's the arachnoid and the subarachnoid space, right? Here's the subdural space. Here's the meningeal layer of the dura mater. Here's the periosteal layer of the dura mater. Does that make sense? Okay. Periosteal layer of the dura mater. What is between them? What is between the many places in your brain are big, wide blood vessels. And they are called the dural venous sinuses. I'm going to show you a picture of them. Ready? It's a cool picture. I love it. The dural venous sinuses. Okay, you ready? Okay, so what am I looking at here? Which layer? Which layer of the dura mater? Meningeal layer of the dura mater. Superficial to the meningeal layer between that and the skull are these big honking blood vessels. These are called, again, dural because they're in the dura mater between the layers. Venous because they're veins. Sinuses because they're big and wide. Okay? And you're going to learn the names of them, but not today. Okay? So these dural venous sinuses, let me tell you what they do so you can figure out why the hell you should care about them. These big wide blood vessels do two things. First off, they drain venous blood from your brain back into your venous system, right? They're going to ultimately dump into the subclavian veins right here. You ready? Okay, so they do that. But they also are nice and wide because in addition to draining blood from your brain, they also drain cerebrospinal fluid from your brain. Does that make sense? So they're big honkers. And pretty much what they're doing is draining this way into your neck. Okay? So those are the dural venous sinuses. They're kind of hard to see in many, many places. I love this dissection. I want to figure out how to do that. So let's review just a little bit. So you're seeing the meningeal layer right here, and this is the dural venous sinuses that are superficial to the meningeal layer. What do you think I'm seeing right here? What layer of the meninges? You're probably seeing pia with a little bit of arachnoid on top. Usually a little bit of the spiderwebby stuff stays on top, right? Not always, but usually. Okay, so those are called the dural venous sinuses. The one that you will find easiest is this one right here called the superior sagittal sinus that's right under the sagittal suture. Does that make sense? So let's go back to this figure that we looked at before. Ready? Okay, so what you are seeing here, guys, is again, meningeal layer of the dura mater, periosteal layer of the dura mater, big freaking blood vessel right there between them. That's why it's blue. It's a big vein. Okay? And that one would be right here. This is actually the superior sagittal sinus, that one. Okay? You don't have to know the name of them specifically yet, but I'm just going to tell you that one. Okay, so the inner layer of the dura mater is the meningeal layer. And that's the layer you are seeing right here, the meningeal layer. And the meningeal layer forms the external opaque covering of the brain. Until you take it off, you can't see a damn thing about the brain. And it extends, so stick with me. This was really hard for me to understand. Maybe it's easier for you guys. I'm not very good at spatial imaging, so like I kind of over-explain spatial things because I have a hard time with them. So here's the deal. You've got this big opaque covering over the brain, right? But the opaque covering actually folds into the biggest folds of the brain. And the biggest folds of the brain are this one right here, called the longitudinal fissure, right here, between the two halves of the cerebrum. You've got one between the two halves of the cerebellum, okay? And then you've got one between the cerebrum and the cerebellum, okay? So that opaque meninx will fold into all three of those. So you're gonna have three big folds. That means that if I pulled a brain out, and I actually wanted to see the structures of the brain, I would have to pull the meningeal layer off, and when I pulled it off, I would have to pull it out from between here, out from here, and out from here. You could flip it over inside out, and you could see all of those folds, okay? So this is showing you the folds. There are three of them that you care about. The other one's really hard to see. I never ask about it. There's actually four of them, but this one right here is a big opaque chunk of the meningeal layer that goes between the two halves of the cerebrum, and it's called the falx cerebri. You've got a little teeny one right there that goes between the two halves of the cerebellum. That's called the falx cerebelli. Do you see it right down there? See it here? Okay. Falx cerebri, falx cerebelli. And then this one is the hardest one to picture because you really can't see what the hell you're looking at unless you have a 3D image. It's the one that goes this direction. That's called the tentorium cerebelli. It goes this direction. Now, here's the good thing though. Next week when you are doing brain in lab and you have the space to think about this, I want you to make absolutely certain that you do not spend the entire time looking at models and the sheep brain. because we have done these cadaver dissections so that you can see. So you probably haven't opened up the cadaver's cranium yet because you didn't have any reason to. But at least 2 cadavers on each campus. We've done the dissection so that you can actually take off the calvarium, which is the top of the skull, right? And then we did the dissection so that you can reflect on one side of the brain, you can reflect the dura mater back. And then I made it so that you can take half of the brain out. And when you take half the brain out, you can see all of those. Does that make sense? You can actually see what the hell you're talking about, and it's hard to see when you're just looking at a 2-dimensional picture. So, on both campuses, there was also a dissected sagittal head that's just separate. So what I want you to do next week is make sure you roam around and look at what's available so that you can actually get the idea of what we're doing here. Besides, those are really cool dissections. They're super fun to do, and they're really hard. So now, the last thing is that this falx cerebri actually attaches the dura mater to the skull. So when I take the brain out, if I want to take a whole brain out, I actually have to cut it off because the falx cerebri is attached to the crista galli. Remember the crista galli? Crista galli of what bone? Yeah, ethmoid bone. So it's attached to the crista galli of the ethmoid bone. If you want to pull the brain out, you actually have to slice it off the crista galli. So it's like holding it in there. Does that make sense? So it's attached to the crista galli of the ethmoid bone. So, and I don't ever ask you about this one. The diaphragm ocelli, because there's just like almost no place that you can figure out what the hell it's talking about, but this is basically the location right here. There's a little chunk that goes right that direction, but I won't ask you about it because I can't ever find a picture that shows it anyway. Now we went through dura mater, and now we're doing subdural space, but there's really just a little bit of fluid and sometimes some blood vessels in it. And then we go into the arachnoid. The arachnoid has some more detail. Let's look at the arachnoid again. Again, here it's represented as gray, and then you can blow it up here. I'm going to tell you something important about the arachnoid. The arachnoid is where the cerebrospinal fluid circulates on the outside of the brain and the spinal cord. It's also on the inside of the brain and the spinal cord, but this is important. Notice that the arachnoid and the subarachnoid space actually reaches up into this blood vessel. Do you see that? Okay. This little place where it reaches up into the blood vessel is a one-way valve that opens in response to pressure. So what is going to happen is as the fluid builds up down here, it's going to open that valve. And this blood vessel is super low pressure because A, it's venous, and B, it's wide, right? So it's going to be super low pressure. So as the pressure starts to build up in the subarachnoid space, the cerebrospinal fluid pressure, it will open that up and dump excess cerebrospinal fluid into the bloodstream. So that's how you get rid of it. That's how it gets out. Okay, we'll talk about how it's made in just a second. So you dump excess cerebrospinal fluid. This structure right here is called an arachnoid villus. Plural is villi, right? Arachnoid villus. And you can see that there's more than one of them, right? They're all through here. So this is basically an out valve. Why do we not want it to be an in valve? Do we want things coming back from the venous system? No. We do not want things coming back. It could be pathogens, and at the very least, it doesn't have very much oxygen, high levels of carbon dioxide, high levels of waste products. Okay, so that's the arachnoid and the subarachnoid space. Subarachnoid space is within the arachnoid, and it's the space where cerebrospinal fluid circulates on the outside. In addition, it's not showing it in this picture, but there's a bunch of little blood vessels, not super tiny, that are going through the subarachnoid space. So imagine that you can see a blood vessel running through there too, because that's actually what you're seeing right here. So in this figure, when you're looking at it, these little teeny blood vessels are probably deep to the pia, but this guy right here was within the arachnoid. Does that make sense? This big one? Okay? You know it's got to be deeper than dura because you wouldn't be able to see it if it was in the dura, right? So the little teeny guys are deep to the pia, and this one is within the arachnoid. Okay, so arachnoid villi, we did that. Okay, and then we get to the pia mater, and we've already kind of talked about the pia mater. The pia mater is the deepest layer. It's tightly bound to the surface of the brain. When you do the sheep brain, after you're finished doing all of the dissections you're supposed to, try to take some tweezers and see if you can pull the pia off without pulling the tissue off. It's pretty hard to get it off, but it's worth it. I mean, you already did the dissection anyway, right? Okay, so that's the pia mater. Okay, now, The third line of protection, after bone and all the meninges, is going to be the fluid, right? So the cerebrospinal fluid is a protection of its own. But the cerebrospinal fluid is kind of a complicated story, so you have to stick with me while I explain it to you. Cerebrospinal fluid is made inside the brain, but it circulates both inside and outside the brain. It's a really, really rich, nutritious juice, and you don't have much of it, but it's going to circulate in and out of the brain. Now, I've told you before, but let me reinforce that your brain is actually hollow. So it actually has hollow spaces within the brain. Those hollow spaces within the brain are called ventricles. So let me show you what they look like. So here are the ventricles of the brain. I'm going to teach you the names of them and a little bit more about them. But do you see these hollows within the brain? There's 1, 2, 3, and then a little passageway that leads to the fourth one. Okay? These are called ventricles. They are continuous with the hollow in your spinal cord called the central canal of the spinal cord. You with me? Okay, so the ventricles, there are 4 of them. You can either number them 1, 2, 3, and 4, or you can just call these 2 the lateral ventricles because they're on the sides, lateral ventricles, because nobody ever remembers whether the right one or the left one is 1 or 2. So the lateral ventricles are 1 and 2. Then this one that looks like a chicken head right here, okay, that is between the 2 halves of the brain right there. That is the third ventricle, yeah? And then it leads through this narrow little aqueduct to the 4th ventricle, and the 4th one is between the cerebellum and the brainstem. Does that make sense? Okay, so one more time. 1, 2, 3, 4, or 1, 2, 3, 4. Okay, these are the hollows within the brains. Lining the hollows within the brain are the cells that make cerebrospinal fluid. I just told you those. Who remembers which one they are? Which cells make cerebrospinal fluid? Yeah, ependymal cells. Does that make sense? The ependymal cells. So lining these spaces are what look like epithelial cells but aren't. They're the ependymal cells of the brain. So you saw those up here, right? Right here. Okay? So I want to teach you a little bit more about how you make cerebrospinal fluid in just a second. First, let me tell you what the fluid is like. So we've got the ventricles, we've got the central canal. What is the fluid like? And then I'll come back and pick up how it's made. You only have about 500 mils or about half a cup or so of cerebrospinal fluid at any given point. But if it varies a couple of tablespoons either way, you are going to be in hell. You're going to have the world's worst headache. So it's really, really tightly constrained, both the volume of the cerebrospinal fluid and also the composition. So what is in it? What's in it? It's basically like blood plasma with all the best stuff in it. Disproportionate amount of glucose, disproportionate amount of vitamins, amino acids, boatloads of oxygen in it. And what you are doing is you are bathing the inner portion of your brain and the outer portion of your brain with this really nutritious fluid. In addition to being like chemically protective of the brain, it's also like your brain is floating in it. So notice that your brain, on a good day, if you got smacked in the head, your brain is not going to smash up against your skull, right, and get a bruise, which is called a concussion, right? It's going to sort of float within the cerebrospinal fluid, which is right here in this gray space right there. So it's cushioning your brain from jolts. Of course, you can get concussions, but you don't get as many as you should. You bumped your head a whole bunch of times without getting a concussion. So it's sort of cushioning it there. So let's talk about how it's made. So this picture in your textbook is pretty poor, so I'm going to have to do a lot of describing. So let's look at, okay, so in the hollows, lining the hollows of your brain, you're going to have these little structures that look kind of like boogers. So look, do you see these? These are the 2 hollows of your brain, the lateral ventricles. And these are these little pink boogers on the walls of the lateral ventricles. Okay? So what those are is ependymal cells with a big wad of capillaries. And what they are going to do is the ependymal cells are going to pull stuff from the capillaries and dump them into the space. When you put the ependymal cells plus the capillaries, it makes this pink chunky tissue on the wall called a choroid plexus. And a choroid plexus is what makes cerebrospinal fluid, right? It's the cells plus what you pull the cells from, or pull the cerebrospinal fluid from. So this is kind of trying to show you it, the same thing, and then I'll show you it from a different view. Here's a capillary, yeah? Here's the cells that are sucking the stuff out of the capillary, and here's where you dump it into the ventricle. Does that make sense? Okay, so if you look at the same figure that we've looked at before, you will see the choroid plexus. So look here. So where do you make cerebrospinal fluid? In all of the ventricles. 1, 2, 3, and 4. So look. You can't really see 1 and 2 in this picture, can you? Because they're lateral to here, although you're seeing a window into one of them. So do you see the pink boogery stuff on the wall? That's choroid plexus making cerebrospinal fluid, right? We had more on this side, more that you couldn't see on this side, yeah? And then notice that there's some on the wall of 4 as well. Do you see that? That's the choroid plexus where you make cerebrospinal fluid. So this is important because sometimes it can go wrong on you. So what is going to happen is everything that was made in 1 and 2 can't leave the brain unless it goes through 3. Everything that was made in 3 can't leave the brain unless it goes through that aqueduct and out 4. So there's only one out path out of the brain. You ready? Everything that was made here in 1 and 2, and you can't see 2, has to go through 3, and then you make more cerebrospinal fluid, right? So you got everything from 1, 2, and 3 that has to go down this aqueduct called the cerebral aqueduct or mesencephalic aqueduct, and then you make more in 4, okay? And the only way that you can get out of the brain is out down the central canal of the spinal cord. You can push it down that way, or there's some holes that lead out from 4, and they're called the apertures, meaning holes, right? Does anybody take pictures, apertures, camera? Okay, so the median and lateral apertures. So let me show you the median and lateral apertures here. They're at the base of the brain. They're really like right here, 1, 2, and 3 at the base of your cerebellum. But let me show you a different picture that shows you where they are. Okay, so median and lateral apertures. Here's one. Okay, here's one. That makes sense? And then you would have another one on the other side. Okay, 1, 2, 3. Okay, so where do those lead from and where do they lead to? They lead from the inside of the brain, from the ventricles, into what space? What space has cerebrospinal fluid? Subarachnoid space. Yes, so they are leading into the subarachnoid space. So now what we can see is that we are going to have cerebrospinal fluid 2 distinct places, within the brain and outside the brain. Yes? Okay, so they are going to lead into the subarachnoid space. It is all circulating, and here is something fascinating about cerebrospinal fluid. Unlike your other body fluids, in which if you make too much, there's a feedback mechanism to stop you making them, there is no feedback mechanism that can stop you making cerebrospinal fluid. You make it continually throughout your life. Logic question for you. Where is the newest cerebrospinal fluid? Inside the brain. Where is the oldest? Outside the brain. How do you get rid of the oldest? As the pressure starts to build up here in the subarachnoid space, what does it do? Dumps it back into the bloodstream. Does that make sense? That has to be continuous turnover all the time. Why do you not want old cerebrospinal fluid? Because it's not going to have very much glucose, it's going to have carbon dioxide in it, not enough oxygen, used up the ions, the nutrients. That makes sense? So you have to have a continuous turnover, which is both good and bad. It's fine as long as it's working properly. If it doesn't work properly, you are screwed. So let's see what happens with that. So here's CSF circulation. We've done it once, but we're going to follow it the whole way this time. Okay? Cerebrospinal fluid circulation. I'm going to tell you right now that you are going to be asked cerebrospinal fluid circulation on your test. Okay? And I want you to trust me about that. You are going to be asked cerebrospinal fluid circulation on your test. You ready? So here's what we're going to do. We're going to go through it in a flow chart, this form this time. We're going to make sure we make it all the way from the sites in which it was produced back to the bloodstream, right? Tell a complete story. So remind me again, what kinds of cells produce cerebrospinal fluid? Ependymal cells in choroid plexus. So we start the ependymal cells of the choroid plexus of the lateral ventricles, which is 1 and 2, right? Everything from 1 and 2 goes to 3, and then we make more and the ependymal cells and the choroid plexus of the third ventricle, right? And then everything from the third ventricle, all three gathers and has to go through the cerebral aqueduct. Keep the cerebral aqueduct in mind. Here it is again. It's the one way out for everything that was made in three different ventricles. So the cerebral aqueduct, and then it goes into the fourth ventricle, and then you make more, right? You make more with the ependymal cells and the choroid plexus of the fourth ventricle. And then from there, there's two different fates. You can either push it down the central canal of the spinal cord. If you do, most of it actually just gets pushed back up. Some of it actually leaks out down the bottom. And then, or you can go out the median and lateral apertures. If you go out the median and lateral apertures, you're going into what space? You're going into the subarachnoid space. The subarachnoid space, remember, is continuous all the way around the brain and the spinal cord. Circulates all the way around both. Okay? Go into the subarachnoid space. But then, how do you get it back into the bloodstream? The primary way is that it goes out the arachnoid villi, those one-way valves, back into a blood vessel. Let's say the superior sagittal sinus or one of the other dural venous sinuses. That make sense? There's one other way that they figured out just recently. I don't usually ask you about this one. They actually figured out that some of it actually leaks out the subarachnoid space down your nerves that leave your brain and your spinal cord. So a little bit of it leaks out that way and then goes back into your lymphatic system. So now, this means that if you get a blockage and there's no mechanism to stop making cerebrospinal fluid, you have got a problem. But if you have a blockage in one of the apertures, it can just leave out the other two, right? Where's the one place that a blockage would be really, really bad? Cerebral aqueduct, right? Because you're not going to stop making fluid in the other meninges. If that occurs and you're still during development, it's in utero, then the baby will have hydrocephalus. This is relatively severe hydrocephalus. So what is the problem with hydrocephalus? The problem is that generally, it doesn't have to be the cerebral aqueduct. That's a very likely place for it to get blocked. You have a fluid that's building up where? Inside or outside the brain? Inside the brain. If it's blocked the cerebral aqueduct, it's building up inside the brain. And it's putting pressure on a developing cranium, right? And then what's the cranium going to do? Well, the bones do. They respond to what you tell them, right? It's going to increase in size. But it's also putting pressure on a developing cerebrum by smashing it up against the skull during development. So what, with this level of hydrocephalus, is almost certain to be true about this baby's mental development? Yeah, you are going to have permanent effects on the mental development of a baby with this level of hydrocephalus. Now, you can also have minor hydrocephalus, in which is just a little bit of an increase in pressure, right? So this baby probably had some developmental delays that were relatively serious. I don't know about this baby. So I want to show you what is actually happening inside. This is not from this baby, but look. So remind yourself right here. So look at the size of these two lateral ventricles, just for comparison, right? So these are two lateral ventricles. And this is in a relatively normal adult brain. Now I'm going to show you a post-mortem photo of a coronal section of a baby's brain that had hydrocephalus. And look at the lateral ventricles there. Do you see those lateral ventricles? Yeah. So this is major. You can tell that it actually got caught up there. So let's talk just briefly about why the hell they don't go in and try to fix this and how you actually diagnose it. Hydrocephalus is not that uncommon in babies. So does anybody in here have kids but me? Anybody? 1, 2, okay. So there's a few of us. Okay, so when you are pregnant and you are getting maternal care, one of the things that they'll start doing is when you go in for a sonogram, they're actually going to do some measurements, and they measure the size of the baby's head. They don't just measure the head, because it doesn't tell you anything. Maybe I just have a big-headed baby, and I do. You know, a little blockhead, it's adorable. So what they also do, they measure the size of the head, but they also measure it with reference to the humerus and the femur and a couple other bones, right? So is it just a big baby, or is that an abnormally large head, right? Okay. So then what do? If, for instance, you figured out that you had a baby that had hydrocephalus, what can you do? Well, mostly what they're going to do is they're going to tell the parents that your baby has hydrocephalus. And so why do you not do, heroic measures to intervene and actually do shunts? First of all, those surgeries are really, really unlikely for the baby to survive the surgery. And second, when you can tell the baby has hydrocephalus, what is probably also already true. Whatever damage has occurred during the development of the nervous system has already occurred, right? So why measure at all? Well, sometimes you can't get vaginal delivery with hydrocephalus, and sometimes, I know it's an uncomfortable thing to talk about, sometimes if the damage that has occurred is so severe, then the parents have some choices to make. If you have a baby that is not going to live into the third trimester or not going to live for moments beyond birth, then some families will choose to terminate that pregnancy instead of carrying it from 10 weeks to 40 weeks, right? If the pregnancy and the baby has like massive developmental problems that you can see already. So they can't really do a whole hell of a lot with intervention, but they can do something with diagnostics at that point. But minor hydrocephalus, a baby can totally survive minor hydrocephalus, and sometimes you don't even have symptoms of minor hydrocephalus. But major, they're going to start watching what's going on. So what would you do in a situation of hydrocephalus if it didn't occur in a developing infant, but occurred in an older child or in an adult? Like, for instance, if I had some kind of injury that blocked my cerebral aqueduct. So what do? They actually just shove a shunt up there. They take a shunt and they shove it in one of these holes, right? One of the apertures, and they shove it, a relatively rigid tube, up past the site of blockage, because you can't stop cerebrospinal fluids production. So then what they do with the shunt is they either dump it into a blood vessel, generally a vein, because you don't want the pressure going backwards. So you can dump it into like your subclavian vein, or sometimes they run it under the skin and they dump it into the peritoneal cavity. Isn't that interesting? Because you want to make sure that you're dumping it someplace that's really dependably low pressure, because you never want anything going back, right? And the peritoneal cavity is really low pressure compared to. And then if it's a child and they had a shunt, then as you grow, the shunt has to be changed as it gets longer, right? It gets a different distance, right? In an adult, they don't have to change the shunt unless it gets scarred up or blocked up or whatever. So you can totally survive those kinds of things. And sometimes it's a complication of like an injury, a brain injury. So this was already relatively narrow and it can close if you have some kind of scarring that occurs. So that's hydrocephalus. The pressure from buildup of cerebrospinal fluid in the brain, you have to do something about it or else it's going to cause brain damage. Okay, so now let's talk about the last line of protection. And the last line of protection is both anatomical and physiological. It's called the blood-brain barrier. So I want you to understand what I mean by anatomical and physiological. So we saw this before. So this guy right here, this is a capillary in the brain. And the brain is like really, really super duper vascular. Tons and tons of blood vessels in the brain. And it's a blood hog. It is a blood and nutrient hog. So the brain only accounts in an average weight person for about 2% of your total body weight, but it gobbles up about 20% of your oxygen and your glucose. And the metabolic rate of the brain awake and asleep is hardly different at all. It's really, really busy when it's asleep as well. So it's not like your skeletal muscles, which use a bunch of glucose and oxygen and then a ton more when you're exercising. Your brain is really never resting. It's not. It's just doing different things when you're asleep. And your brain is intolerant of decreased blood flow because, in part, it's really, really energetically expensive and it has almost zero capacity for, get ready for a bunch of syllables, anaerobic cellular respiration. So you know what that means. You tell me. Anaerobic cellular respiration. Yeah, producing what without oxygen? No, it's using glucose to produce ATP. Okay, so it can produce almost no ATP in the absence of oxygen. So this is really important. Constantly gobbling up ATP, really energetically expensive. Can't make it unless you supply it with oxygen. So that means that your brain gets hella pissed if you ever decrease the oxygen supply to the brain. Your brain at normal average temperatures will start having cell death within a 1 to 2 minute interruption of blood flow to the brain. Other tissues are not like that. And of course, is the damage permanent if it occurs? Yeah, so that's generally how strokes occur, right? You either get a blockage or an aneurysm that decreases blood flow to the brain. And with 1 to 2 minutes, you are going to start losing brain cells because of that. So this is at relatively normal average temperatures. Anybody know why, if you had to drown, first of all, don't drown. But if you were going to have to drown, you want to drown in a lake in Minnesota in February and not in a lake in Texas in July. Why?
The cold slows everything down.
The cold it slows down the metabolic needs for oxygen, so your tissues can last longer without it. Does that make sense? So all of your metabolic processes go more slowly at lower temperatures. And in that particular instance, it's really good to go more slowly at lower temperatures. So you've heard of people like, drowning in a frozen lake and being revived after like 15 minutes. It does not happen when the lake is warm, right? Because you slow down the metabolic needs of your brain. Okay, so let's talk about the blood flow to the brain and where it comes from. So most of the blood flow to the brain is through just 4 arteries, which is crazy to think about. So you have 4 arteries supplying all of that blood supply to your brain, and then we'll talk about the blood-brain barrier. Okay, so here are the arteries that are supplying the brain. You already know something about these arteries, even though you don't know that you do. Okay, the 2 pairs of arteries that are supplying the blood supply to the brain, one of them splits off right here, and it's called the internal carotid artery, and it goes through a hole that you learn.
Carotid canal.
Carotid canal, good. Goes up through the carotid canal, which enters like right by the pituitary gland, right up there at the bottom, okay? So those 2 arteries supply about 70-75% of the blood supply to the brain, which is why you never want to play that dumb pass-out game that you did when you were a teenager. Remember that pass-out game? Right? That's what Utah kids do instead of drinking beer. You should drink a beer. It's more healthy for you than a pass-out game, right? Do you know what a pass-out game is?
Yeah.
Right? Like compress your neck to see if you can get that woo, you almost died feeling, right? Honestly, you're a little far less likely. Don't get in your car. Don't have 20, right? These are the internal carotid arteries. 75% of blood supply to the brain. The other 2, you actually kind of met something about them because the other 2 come up here and go through those holes in your cervical vertebra. Remember those holes? What are those called? Transverse foramina. Those 2 arteries are called the vertebral arteries because that's where they run out, right? And they go up those holes and they go anterior to the brainstem and then they come up through that hole, through the foramen magnum. So you got 4 arteries delivering all of the blood supply to the brain. There is some redundancy in those arteries because if you lose one, you might very well survive because one of the things that we'll see in the brain is the blood supply to the brain has a bunch of redundancy of circulation. Like if you can't get it up this way, it'll go all the way around and get there the other way. So these 4 arteries supply all of that blood supply to the brain. And apparently you can live without one of them because I am living proof that you can live without one of them. This guy right here, my right vertebral artery. Very stressful time in my life in which your blood pressure increases. We've all had them, right? Okay. But apparently during that very stressful time in my life, I actually had the combination of high blood pressure, and apparently I have a weakness in the wall of my arteries. And what happened is my right vertebral artery developed a fistula and busted and started dumping directly into my right vertebral vein. Okay. I didn't even know what happened, although I was sitting on the couch and I started hearing so incredibly loud. Like all of a sudden I could hear my arteries pulsing through my skull. So I could hear whoosh, 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 whoosh. And it never stopped. It didn't stop at all. And I didn't go to the doctor even though I know I've got cardiovascular problems because I was thinking I was going to get divorced. And I was like, I'll deal with that after I get divorced. But I didn't get divorced. But I did bust a blood vessel. So it was so loud that I couldn't like sit with you on the right side and hear you talking to me. So when I finally went into the doctor to get it checked out, what he told me is my right vertebral artery had developed a fistula and started dumping right into my right vertebral vein, and I didn't have a stroke. What the hell? right? I just like cut 12.5% of the blood supply in my brain, but apparently there's so much redundancy that I didn't have a stroke. I mean, I might have been smarter before then. I don't remember though, because this is all the smarts I got. What they decided to do, and this is kind of interesting, what they decided to do in that situation once I finally went in, and I was like, okay, doc, what the hell's going on? And my cardiologist, literally, this is what he did. He listened to my heart, he's like, everything's fine. He's like, hey, will you just like listen to my skull for a minute? And he goes, oh shit. You're not supposed to say that as a doctor. So then I went in, and the specialist at the U gave me two options, but he ended up choosing the option. Either they could try to sew it up, right, so that you could regenerate blood flow to the brain, but he was afraid I would throw a clot if he did that. So what he did instead was like, well, apparently you can live without it. They just shoved $30,000 worth of platinum coils up that thing. Yeah, don't kill me. I don't think there's any resale value. $30,000 worth of platinum coils up that thing and shut down the blood vessel permanently. Because what would happen, just think about this. And this is a dumb question that I asked him. I was in a dumb stage of my life. Just give me a break. So I was like, why do I need to fix it? Because apparently I can survive it. Is it just the noise? And he's like, no, basically dumbass. What happens if you dump arterial pressure blood into a vein?
You're putting too much blood.
Too much pressure. Then that vein was going to bust eventually. So he had to like shut it down. So basically, I wonder what that looks like. Well, actually, he let me watch all the way to the last minute. But I wonder what it looks like now. So I donated my body. Somebody's going to get to dissect it out of there. It's going to be fun. Maybe it'll be one of you guys. You'll be like, ooh! Okay, so these blood vessels, so I'm going to show them to you. This is the right vertebral right here going up the transverse foramina. This one right here that's diving not outside the brain, because that's the external carotid artery, but inside the brain, that's the internal, right? The inside of the head, that's the internal carotid. So those four are going to give rise to all of the other smaller arteries in the brain, and you're going to learn some of those small arteries later. But let me just introduce you to what the blood-brain barrier is. So these four arteries are going to split and split and split and split and split, ultimately leading to capillaries in the brain, microscopic capillaries in the brain. And then you guys tell me, these microscopic capillaries in the brain, in order to prevent anything funky from getting out of the capillary into the tissue, what type of cell junction should you put between the cells? Tight junctions, yes. And they are the tightest of tight junctions anywhere. They are called continuous, complete tight junctions. Like, I am not messing around tight junctions, right? Okay, and what these do is they prevent anything from sneaking between the cells to get into the brain, so that the cells themselves can control what gets into the brain. Does that make sense? Okay, so the blood-brain barrier, that in that way, it's an anatomical barrier, right? A physical barrier. But then in addition, the reason that it's a physiological barrier is that the astrocytes can also increase or decrease transport of certain things, right? Or if there's something funky in your bloodstream, they just shut it down. Shut down the transport until you get whatever that funky thing is out of your bloodstream, right? So astrocytes help. So this is a physical barrier, and these help with the physiological barrier. Does that make sense? Okay, so if something has a mechanism to get into the brain, you probably need it. So not surprisingly, you've got boatloads of glucose transport mechanisms. Would you have any like strychnine transport mechanisms to get into the brain? No, because you don't want it in there in the first place, right? So some substances don't need permission. I don't know if you guys know this because you haven't had physiology yet, but lipid-soluble substances do whatever the hell they want. Right? They go straight through any cell into the tissue. So lipid-soluble substances like oxygen, like lipid-soluble substances like alcohol, like nicotine, and anesthetics, they go straight through the blood-brain barrier into the brain. And the blood-brain barrier is really no protection from lipid-soluble substances. But other substances, if you don't have a transport mechanism for it, it is not going to get into the brain very easily. And that's important because sometimes it's good, you didn't want it in the brain, and sometimes it's bad. Like you know you need it in the brain, but the blood-brain barrier won't let it in there. Okay, so dopamine, for instance. So Parkinson's disease, let's back up a little bit. There's a deep portion of the brain called the substantia nigra, and it uses the neurotransmitter dopamine to control voluntary motor activity, okay? With Parkinson's disease, what happens is you lose the capacity to produce dopamine in that region. really because your glial cells aren't supporting the neurons, okay? Well, we know how to make dopamine in the lab, and we know where it needs to go, but we can't get it in there because of the blood-brain barrier. The best that we can do is a lipid-soluble precursor to dopamine called L-DOPA, lipid-soluble precursor, and that works really, really well in the early stages of Parkinson's disease. But in the later stages, after you've lost the cells that can transform it from L-DOPA to dopamine, you lose the benefits of it again. So it works and then it stops working. Does that make sense to you? So other substances can't pass easily as well. Most antibiotics cannot pass easily into the brain. So if you get a brain infection, which are pretty rare, it's hard to get antibiotics in there. It's hard to get most proteins in there. And so the blood-brain barrier actually protects you in a lot of different ways. One of the other things it protects your brain from is from fluctuations in your extracellular fluid. So after I eat, my blood glucose goes up. But if I haven't eaten in a long time, my blood glucose is really down. But I want for my brain to have a relatively constant level of glucose. So what it does is you just increase glucose transport when it's low in the bloodstream and you decrease it when it's high, right? So you make sure that the brain really has like kind of a buffering system for whatever's happening in the body, right? It's just making sure that the brain still gets relatively normal levels. Okay, and then the last thing is, and this is kind of interesting, if you do have a head injury, you have temporarily disturbed or destroyed your blood-brain barrier. But it's epithelial tissue, so it'll regenerate as long as you live through it. And what you can do is, let's say you've got a closed head wound, like you get smashed in the head, something like slightly worse than a concussion, right? The only good thing about that happening is that is also the moment in which you can get antibiotics into the brain. Why? Because you've just destroyed the blood-brain barrier. Does that make sense? So you've temporarily destroyed the blood-brain barrier. So if I had a closed head wound, what they would do is two things at the very least. First of all, they'd try really hard to manage the swelling because there's no room to swell in there. So they'd give you prednisone or steroids or something. And then they'd give you antibiotics, but just in case. They don't really know if anything got into your brain, but this is the time that you would get them in there, right? They call them prophylactic antibiotics, just in case something happens. So they use those to prevent the swelling that can occur. And I've got one last little thing to tell you about the blood-brain barrier, and then we'll do brain next time. Ready? This is a really, really cool, I think it happened last year or maybe a year and a half ago. Some brilliant freaking scientist figured out that you could temporarily destroy the blood-brain barrier in one area and deliver targeted drugs to that area after you destroy the blood-brain barrier. And this is how it works. I'll show you briefly. Okay, so what they did is they actually injected micro bubbles, right? Not big enough to cause an aneurysm, just teeny tiny bubbles into the bloodstream. And then what they did is they shot ultrasound at that little location. So let's say I have actually a tumor in my brain, right? I shoot the bubbles into the bloodstream, right? And then what I do is I shoot ultrasound at the bubbles in one little location. And the combination of the bubbles and the ultrasound temporarily destroy the blood-brain barrier. And I use that opportunity to deliver targeted chemotherapy drugs to that location. Does it go everywhere? No, because the blood-brain barrier keeps it from going to places in the brain that you are not shooting the ultrasound at. Right? And then as soon as you take the ultrasound off, the blood-brain barrier seals itself back up, and then they can do it again. That's freaking brilliant, right? I love that. Okay, I will see you guys on Thursday. No quiz this week. We'll either have one take home or have one on Tuesday if the test moves.